So welcome back to the episode of BioBlast. I have the pleasure of having John Mumm, the CEO and founder of Deka Biosciences, join me today.
Hey, how are you doing, Devin?
I'm doing well. I'm doing well. So excited to connect with you, John. I know you are very busy scaling the company, and we're looking forward to diving into your professional journey and certainly the technology that you're developing at Deka Biosciences. But just start for those that are less familiar. Can you just give us some background in terms of your training, your sort of professional journey?
Yeah, so I grew up in a small town in Northern California, and the folks that I kind of was in the town with, they either built houses or they worked at the supermarket or they logged in the forest, cut down trees, and I did all those things as well as a kid. But what I found was I really had a notion of what was science about, and this is I'm old enough that I was interested in science before kind of anyone thought that was a cool thing. Although I'm not sure that people think it's so cool today, but I do. But I had the fortune of sitting in an assembly at my school, and there was this guy, even forget his name, but he was kind of a technologist, and he said, your job, your profession hasn't been invented yet. And he was talking to the senior class. And I literally sat there and I thought, hot damn, like that's great, because I don't see anything that I necessarily want to do for the rest of my life. Right. And kids are kids. We always, when we're young, we think that, we're going to go change the world. But that was kind of that was an important kind of metanoia for me, that there was something outside of this town. And not to say that I don't like the town. It was a great town. It was an amazing place to grow up. It's just I wanted something else. And I didn't know what that was. And what that conversation permitted me was to start thinking about what else there may be. And so I literally looked at myself. I said, okay, self, like what do you like? What interests you? And I realized that, Mr. Taylor, my chemistry teacher, one of the funniest guys ever, and taught me, the basics of chemistry, and I loved it. Dr. Soleil was, we were lucky enough to have some PhDs teaching high school. So Dr. Soleil taught biology, and he also was a contractor. And I worked with him building houses, and then I met him in school. And I realized again that there's this very interesting, optionality, if you will, to the way we use our brains, the way we use ourselves. And then Mr. Fuji, so for us in physics, F equals MA was Fuji equals mental anxiety. And so, it was obviously wasn't one of my strongest classes, but, that was a whole eye-opening experience as well, in the sense that there's this massive spectrum of scientific endeavor that is open to us humans. And so I actually stayed out of college for a year. I was a runner. I had an opportunity to run in a bunch of places, but I stayed out to help my parents start a business, and I did. And then when that year ended, I went to school, and I got myself into a very small, as it turned out, liberal arts school, Menlo College, down in the Bay Area. And there, I was actually a humanities major for 2 years. Because I really didn't know what I wanted to do. I loved writing. Again, Fuji, had scarred me sufficiently that I knew that I wasn't brilliant at physics, but I still, kind of was a dual major. I had what was called a biotechnology degree and a business degree. And my school was actually the first one that offered a biotechnology degree. It was a brand new field. And in fact, my very first week in college, we had this freshman dinner seminar series, and the founder of Core Therapeutics, who's now a world-renowned VC, came and gave a talk, and he told us about what is biotechnology, and at the time the barriers to entry were impossible, right? I mean, he had raised, I don't know, $50 million, and they were making small molecules. He was literally having to invent the entire process outside of pharma in terms of how you do that. Again, the very beginning of biotech. And then for my degree, I actually had an internship requirement. So as I said, I was a runner. I went to school to run. My very first season, after my first cross-country season, I got in a massive bike wreck and did nerve damage to my leg. So I could no longer run. And again, I was like, what are you interested in? And I was like, I really love science. I love, crazy as it sounds, I love organic chemistry. I love, in particular, protein biochemistry. That was really something that I could see, I could understand. Working in the lab, even at our small school, I was back to kind of working with my hands and my brain at the same time, very similar to what I'd done with my father in terms of building houses. So super interesting. And what I started doing was working full time. I got a job at a marketing place for semiconductors because we were in the Bay Area. That was really interesting. I hung out with the scientists looking at all these different ways to accelerate kind of cesium ions, and then some of the first time of flight analysis for proteins was being done there. Again, kind of this theme of being interested in proteins. I then needed a bit more money than that job gave me, so I got another job in recruiting. So I was in biotechnology, pharmaceutical recruiting for executives. And that was another kind of path for me to see, looking at these CVs and resumes, what it meant to be an executive in biotech and in pharma. So again, kind of mapping my path as I went forward. And then finally we get to the internship, and I was hired before I actually finished my degree at this company called Anergen. And at Anergen, we were trying to energize, it's an old word, we were trying to downregulate the functionality of autoreactive T cells. And I was kind of in this company trying to suppress the immune system that was inappropriately reacting to self. And I thought, gee, cancer is self, and wouldn't it be interesting if we could activate the immune system to destroy tumors? Now, at the time, this was heretical in the sense that everyone was working on, kind of apoptosis, what had just been discovered, programmed cell death. Everyone was working on cell cycle, building small molecules to suppress the kind of proliferative cycle of tumor cells. No one ever at the time thought that the immune system could ever be sufficiently turned on to destroy self. But for me, 8 members of my family had died from cancer. And so one of my earliest memories is my grandmother coming home bright red because of full body radiation. And so that left a mark, as it were. My mom and my aunt died on both sides of my family. My, uncles died. I mean, it's, my grandfather died. So, a lot of clear, clear awareness of the standard of care at the time, and the potential promise of biotechnology, nascent as it was, to really impact people's lives in a positive way. So again, I was studying for the MCATs, and Dr. John Farah, the CEO of Anergen, came in, and he's like, what are you doing, John? And I was like, well, I'm not sure if I want to go to med school or grad school. And he goes, you're going to go to grad school. And I was like, well, how do you know? He's like, I know. I just know these things. I was like, okay, whatever. So I still thought I wanted to go get an MD. And so I actually got into a program that was a master's MD program at Boston University. And so I would do my master's, do well enough, and then I would matriculate into the MD program. And my wife and I got pregnant, and her family was there in the Bay Area. And she was like, there's no way I'm moving to Boston, with a child. And I was like, okay, so I clearly need to get into some program closer to home. So as it turned out, Stanford had a master's program that would permit me to take kind of all of the graduate level courses in any of the science. Clearly I have issues with, kind of choosing a career, but in any of the sciences, as well as in the med school. And so I interviewed with, Nobel laureate Dr. Paul Berg and a few others, and they're like, okay, you don't sound like you're completely insane. You can go ahead and come to Stanford to do a master's. And I was like, hot damn. And I needed to work, right? So had a baby on the way, and you have to pay for your master's. So one of the scientific advisory board members for Anergen was Dr. Mark Davis, so kind of super famous T cell guy, protein biochemist as well. And I called him up and I said, hey, I just got into a master's program. I need a job. Do you have an opening? And he said, wow, this guy's, got some chutzpah, sure, let him in. Let's see what he can do. So I started my master's and my new job on the same day. And Mark did me the amazing service of making me an HHMI employee as well. So I kind of got a sense of what that part of academia also means. And so for the first 3 months, no one talked to me in the lab, right? Because I was a tech, and I guess you don't talk to techs. And I was actually in class, and so Mark's wife, Yusha Chen, I was taking her class, and the Mark's lab had a lab meeting, and I come back from class, and suddenly everyone's like, "Hey, John, like, how you doing?" And I was like, "Oh, I'm doing fine. Have I been invisible for these past 3 months? Like, what's going on?" They're like, "No, no, 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 no. Listen, the work you're doing is amazing." I was like, "Okay, that's great." Didn't really think it was more amazing or less amazing than anything else. But as it turned out, what I had managed to do, so John Altman when he was postdocing in Mark's lab, had made these molecules called tetramers. And tetramers are essentially an MHC with a peptide that is then stuck to kind of a carrier, it's streptavidin. And then you can label that up, and it becomes a reagent to bind to antigen-specific T cells. Now, I had been doing similar work at Anergen, and that's partly why Mark, was willing to bring me in, because he had a need for a protein biochemist that could make stuff. And what had ended up happening is that in that first 3 months, I worked with Dr. Peter Lee, hematologist, and Dr. Mario Schroeder down in Len Herzenberg's lab. Mario had made a 9-color flow. And so what we had done is I had made a bunch of different tetramers with different antigens, peptide antigens to tumors that no one else had really been able to make. And Peter got the blood and Mario ran the flow. And we found, interestingly, that cancer patients, at least the ones we tested, had tumor antigen specific T cells that were of a memory phenotype. So what it meant is that the immune system was capable of recognizing tumors, just something else was going on that was shutting down that system. So, even though, immuno-oncology wasn't a thing yet, this is back in 1997, 98, 99, right? So this is long before checkpoint. This is long before any of that idea. Although Jim Allison was already working over there at Berkeley on CTLA-4, et cetera. So that it was starting, right? That whole notion of the ability to impact the interface between the immune system and the tumor. But I saw what I saw. And Peter published in Nature Immunology, I believe. So, well-received manuscript. And I made up my mind kind of then and there that as Dr. John Farah said, that I was going to go to grad school. So I was sitting in the lab on a Saturday. Mark came in, and I had applied to grad schools. I applied to Harvard, and applied to MD Anderson, and a few others. And he said, "Hey, look, I just talked to the dean at Harvard. Are you sure you want to go there?" And I said, well, why are you asking me? And he goes, "John, you have a kid, and I know you. You're not going to fit in. You're not going to fit in because you're going to want to do your own thing. You're not going to want to do what people tell you to do." And I was like, "Okay, thanks, Mark, I think." And I said, "Well, I have been accepted to MD Anderson as well." And he's like, "You should go there. Like translation, bench to bedside, doing the things that really, really fire you up. That's what you should do." And I was like, "All right, fine. I'm not going to, deny, Mark. I mean, the guy's wickedly smart." So decided to go to MD Anderson, and he throws me a party, going away party. And he pulls me aside and he says, "Look, I got to this place in my career by doing things that no one else has done." Right. And so when I have an observation, it doesn't actually matter if I'm right or wrong. It just matters that I'm the first one to do it. And he goes, "You're not that way, John. That's you. What you want to make therapeutics, so you better be right. You need to be the first one there, but you also need to be right." I was like, "Okay, that's fair." And so I went to Liz Grimm's lab. So Dr. Grimm worked with Dr. Steve Rosenberg, right? So first guy to put IL-2 into people, Dr. Grimm, and helped him do that. First guy to really experiment with TIL therapy and CAR therapy, et cetera, right? So he really, from the IL-2 perspective, started the entire immuno-oncology, path. He was just way earlier than anyone else. And so Liz literally had IL-2 from Cetus Chiron in her freezer, and you couldn't go into Liz's lab without doing an IL-2 experiment to really show that you could sling T cells around and you could work with proteins, et cetera, et cetera. So all good, passed the test, got in, and I then worked on kind of forwarding the idea that we had at Stanford that there's something in the tumor that's shutting down an activated immune response. And so what I had access to in Liz's lab through Dr. Nancy Poindexter and then Dr. Jack Roth, so Liz's husband, he's a surgeon, I had access to patient tissue that I could pull out T cells and tumor cells from the same person and grow them, and then kind of assess the ability of the T cell to kill, to induce cytolysis of the autologous tumor cell. So Nancy was the one that kind of put all of that together. I used a bunch of those reagents, and I asked myself, of the cytokines, right? So remember, I'm a protein biochemist. I like proteins. Of the cytokines that are known to be in tumors, there's no one of them that is the grand architect of immune activation or immune suppression. So if that's true, can I set up a screen where I can combine cytokines that are thought to be in tumors to see what the combinatorial effect on T cells are? Now, then, as we talk about Deka, I haven't left that theme for 30 years.
Right.
In the sense that, I always say that to me, the immune system and the cytokines are the one way to think about it is as if you're sitting in the audience at a ballet and you're watching the dancers dance the story, right? Because a ballet is always a story, but you're not listening to the symphony. But the symphony and the notes that the symphony is playing actually are the instructions to the dancers as to where they need to be in a given time of the story and how they need to interact. It is the music that dictates the dance. And so to me, the cytokines are the notes of the music and the dancers are the cells. And so that's kind of how I think about what I do, if you will. So I set up the screen, and there's TGF-beta and there's IL-6 and there's IL-10, because these are essentially the cytokines at the time that were known to be in patient tumors. And I inadvertently find that the more IL-10 I add, the more activated the T cells become. And I actually thought I did the experiment wrong. I thought, John, you're an idiot. Redo it. So I do it again. Come back 5 days later. This is in between Christmas. And so I give myself a terrible Christmas present 2 days before that it looks like I screwed up this really laborious and challenging assay. I set it up again and then come back in between Christmas and New Year's. And lo and behold, it's the same result. The more IL-10 I add, the more activated the T cells are. Now, the thing to keep in mind is that when you carry T cells in a dish for a long period of time, you have to use IL-2. So I was actually testing the combination of IL-2 and IL-10 together on CD8 cytotoxic T cells. Okay, that is now my lead asset that I'm dosing patients with, is an wild type IL-2, a high affinity IL-10 combo. Because I and others, there's 6 other groups that have published over the years that the combination of these 2 cytokines in particular drive a much higher degree of functionality and survival, et cetera, of the cells that are most capable of killing tumors. So that started then. And I took that whole story through my orals. And Dr. Steve Ulrich stood up and said, because I said that more IL-10 is better for T cells. And he goes, do you realize, young man, that there's 10,000 papers that say you're wrong? And I said, yes, Dr. Ulrich, I know that. I've read most of them. And there are nuances, right? Science is about nuance. Science is about understanding your system and then figuring out how it's applicable to, if you want to make therapeutics, to the whole body. So I said, yes, thank you very much. He wasn't on my thesis committee, but I managed to get through my orals. And so I then pursued IL-10. So I kind of did my thesis on IL-24 because that was what Liz was working on, and I worked full time through my PhD for Sunil Shada over there at Introgen, and he was working on IL-24 as well. So I just kind of had to double stitch what I was doing because by this point I now had 2 children, not 1. And my wife was doing her MBA at Rice as well. So we kind of needed to eat and have a roof over our heads. So, and I've always, up until my postdoc, basically from, second semester, freshman year, all the way through my master's, all the way through my PhD, I've always worked full time, and I've always tried to work in biotech. And the reason is that, I tell my kids, you learn by doing, and especially something like biotechnology. It is as much art as much intellect and as much just brute force hard work altogether, right? And so it's kind of why I'm attracted to this industry, because it's a continual heavy lift, whether it's emotionally, mentally, or physically. It's always a grind. And so ultimately, you learn more from it. I think you have a kind of almost extraordinarily productive life if you can move the ball forward. So when I went to get my postdoc, I went to all the people that had ever worked in IL-10 in the U.S. And as it turned out, I kind of knew some of them. So Dr. Maureen Howard, I'd actually worked with her at Anergen. Dr. What's it, Moore, Kevin Moore, Dr. Kevin Moore. I had interviewed with him, and he was at another company. And I told him about my data, and he had actually discovered IL-10 when he was at DNAX. And he goes, John, this is really interesting. This is actually similar to some of the stuff that we had seen in mice. So yeah, you should pursue this. Unfortunately, it's not going to be here because I'm not working on IL-10. So I was like, okay. Maureen was at Chemocentrics with Dr. Tom Schall. She was like, yeah, John, this is really interesting, but we're not going to do it here. So I was like, okay. So kind of all roads seem to lead to DNAX. And DNAX is, think tank started by 3 Nobel laureates from Stanford. So back to Stanford, back to the Bay Area, again, back closer to my in-laws. And I decided, I'm not making any headway saying that I want to work on IL-10. I'm just going to take whatever job I can get at DNAX and go from there. So I did. And so I was hired to work on a subunit of IL-23. So again, cytokines, subunits P19. And I actually found that, and I worked with Dr. Martinoff, and I found that P19, interestingly enough, outside of the immune system, in context of tumors, is actually expressed on the surface of tumors. So then this is right about the time when PD-1, PD-L1 was being discovered. And so that the cytokine interface and then the tumor cell interface suddenly started becoming extremely important. So, but I still wanted to prove if IL-10 could actually be useful. And as it turned out, they had IL-10 and they had pegylated IL-10 because that Schering-Plough had actually dosed 3,000 patients with IL-10 to look for its anti-inflammatory function. And they found something very interesting. And that is that at low doses, the patients would undergo remission with patients that had Crohn's or psoriasis. But as soon as you started to titer higher than about 10 mics per kg, patients would lose that anti-inflammatory function. And 20 mics per kg was worse than 10. So a gentleman that I hope to someday meet, his last name is Tilg, he managed to get his hands on the patient samples from Schering and evaluate the cytokine levels at all those doses. And he found something incredibly interesting, and that is that at all doses, all dose titrations, IL-10 is anti-inflammatory to a certain cell subset, essentially the macrophages. So it suppresses TNF, IL-6, IL-23, kind of you name the interleukin or interferon. IL-10 suppresses it at all doses except for interferon gamma. And I knew that IL-10 stimulation of CD8 T cells induced interferon gamma. So suddenly I had clinical data that at 10 and 20 mics per kg, IL-10 actually activates human CD8s to cause them to secrete interferon gamma. Now, the reason this is important is that whole PD-1, PD-L1 circuit is driven by gamma. And antigen presentation, class 1, class 2, is driven by gamma. So suddenly I thought, okay, this translation of human T cells with human cytokine in a relevant system seems to recapitulate something that we see clinically, right? When they weren't looking for it, right? This was a completely unexpected observation. And I thought, this molecule can be used in immuno-oncology. And so, as you can imagine, IL-10 was discovered at DNAX. Everyone there was dyed in the wool. IL-10 is an immunosuppressive cytokine, right? And I said, well, it's anti-inflammatory, but it's not immunosuppressive, right? Nuance. Again, understanding the system in all of its elegant complexity. So at DNAX, when you postdoc your first year, you undergo a review, essentially you give a seminar to the entire company. And if you pass that, then you can continue. And if not, then they ask you to leave. And so for my first year seminar, I actually presented two programs. I presented all the work that I had done on anti-P19, characterizing its secretion by tumor cells, characterizing the nature of the molecule. It's actually a quaternary kind of structure, or excuse me, it's a four, how do you call it? So it's four monomers in the same structure. So it's like a dimer of dimers that's held together by a divalent cation. And it's actually very immunosuppressive. Interestingly enough, it's secreted by tumors. And I also presented an entire program on the exploration of the utilization of pegylated IL-10 in mouse studies to control tumors and how it does it. Essentially, it uses CD8s, it induces gamma, it upregulates antigen presentation, all the stuff that would have been inferred from the in vitro work that I did in my graduate work was then proven out in a systems biology approach with regard to PEG10 in tumor bearing animals, and so that led to a cancer cell paper. So for that first year, they did not ask me to leave, but I was rather interested in becoming a scientist, and so I went over across the bay to what was Protein Design Labs and got a job offer. And then I came back, and as it turns out, everybody in the Bay knows each other. And so Emma Lees, who was my boss, had been called by Amy Law. And Amy said, "Hey, I just gave John a job offer. Here's what it looks like." And when I came back, Emma had a job offer for me that looked identical to what I got from Amy. So, or rather from Debbie. So all good. I decided to stay at DNAX, but I was one of the few postdocs that had actually ever been hired as a scientist at DNAX. And there's kind of a short list, because they were pretty exclusive group, because they did great science. And I'm really proud to say that I was there for another 4 years, pushing the PEG10 program and others forward. And we got it to an IND discussion stage, right when Scheer and Merck were doing their reverse merger. And I looked across the table at my new Merck colleagues. And at the time, they were all small molecule folks, right? Anti-PD-1 was not a thing yet. In fact, Andreas von Elsis, who was in Jim Allison's lab, had worked with Wiebe Olive at OS, Organon, and established kind of a small biotech as an offshoot of Organon in Cambridge, Massachusetts. And Andreas actually made all of those anti-PD-1s, the one of which became Merck's, because Scheer purchased Organon, became Keytruda. And so, or Pembro, which I always forget the trade names, but basically became Merck's anti-PD-1. And the interesting thing is, the PEG10 was on the out-licensing docket, and that anti-PD-1 was on the out-licensing docket as well. Martin and I formed a company called Targenix to in-license the PEG10, and Andreas and Wiebe formed a company to try and get the anti-PD-1. And it was right when Roger Perlmutter came in from Amgen to take over there at Merck. And so Martin and I managed to get the PEG10 literally 2 weeks before Roger closed the door on all out licensing. And the anti-PD-1 happened to then not be out licensed, and they developed it. So Martin and I then brought in Peter Van Blesler to form ARMO. And then the three of us, as founders of ARMO, pushed that forward. I ran CMC, even though I knew not a whit about manufacturing, and the preclinical research. And we got, I don't know, 4 or 5 papers there and a bunch of patents, et cetera. And I learned a lot about manufacturing. I wouldn't say I'm an expert, but I did 28, at scale GMP campaigns, and I learned a thing or two about how to manufacture proteins. So we got our first $1 billion term sheet, and 4 years to ARMO. And I kind of looked at it and I said, I learned everything I could at this stage. And my oldest had just gotten into Princeton, and I wanted to be closer to her. So we moved here to the East Coast, and I took a job at MedImmune. So interestingly enough, MedImmune actually gave Martin and I our first term sheet for Targenix originally, and that is what nucleated the interest of Bess Seidenberg and Peter Van Blesler to start ARMO. So came to MedImmune, and I unfortunately saw the demise of all of the co-stimulatory, OX40 and CD137 and ICOS, et cetera, that folks were not just MedImmune, but kind of everyone was trying to develop. And I realized two things. First and foremost, that interaction between, say, OX40 and OX40 ligand or CD40, CD40 ligand, those are molecules that are on the surface of two separate cells. And they are kind of tethered in such a way that they, as soon as they bind to each other, they help to pull the cells together, right? So there's a motive force on either side of that interaction that is kind of a tug of war. Now, if you try to drug that interaction solubly, where you don't have a cell on the other side, the receptor or the ligand actually gets internalized. And so it's a self-attenuating process that actually obviated the utility of those types of drugs in that system. But I realized that the signal transduction of a lot of those co-stimulators are essentially identical or super similar to cytokines. And cytokines do, in fact, function solubly. They're soluble ligands. So I thought, okay, I can't not start another company. And this part of the reason I came to this area, or came to the kind of Germantown, Gaithersburg area, is that there's a huge pool of scientists here. They're very well trained, but they don't necessarily know what biotech is or what it can be. But they're curious. And so I thought, okay, I kind of know how to start companies. What's the idea? What should we do? And I thought, I know cytokines. We also know the liabilities of cytokines, meaning cytokines alone don't really seem to do the trick. And cytokines have disparate biologies. Is it possible to blend natural functions that we know are true in humans together in order to actually make a therapeutic? Now, if you're going to go to the trouble of combining cytokines, why not stick a targeting system on it? Because we also know that if you can get cytokines at high concentrations in tissue, like they usually function, right, in a site of inflammation, et cetera, they actually have a much higher therapeutic index. But I thought, okay, let's see if we can make a trivalent molecule that has a targeting domain, has two cytokines, and is manufacturable. So we kind of started this from the very beginning, from my CMC experience and my protein biochemical experience, to try and make a therapeutic. And so, as I always try to do, I try to make it as hard as possible up front. So I chose wild type IL-2. There's been billions of dollars that have been put into muteins of IL-2. I was unconvinced from the very beginning that was going to work. In fact, John Langowski postdoced in Martin's lab at the same time I was there. Liz Grimm's first postdoc was Laurie Owen-Schaub. Laurie Owen-Schaub was John's PI. And then my PI was Liz Grimm. So John and I kind of have been in the same circles, and then we ended up postdocing in the same place. He then went to Nectar and was instrumental in developing Nectar's Nectar 214. And then he left. And so I went and had dinner with him. And I was like, hey, John, why'd you leave? And he goes, that molecule doesn't have legs. And so that was before anybody knew that the molecule didn't have legs. And so, kind of the more information you can have before anyone else, the better off you are.
Yeah.
And so I already kind of had an expectation that pegylation, that a mutein of IL-2 wasn't necessarily going to work. But we had done an experiment of dosing PEG10 and IL-2 in mice, because IL-2 can be very toxic in mice. And we found that you could dose a lot of PEG10 and block the toxicity of IL-2 in mice. So I thought, okay, here's a chance to really interrogate the system to see if we can rescue, if you will, or if we can live up to its promise. So we made a couple hypotheses. One was that IL-2's toxicity was not, firstly, vascular leak. Firstly, we think that it's CRS. So IL-2 is very pro-inflammatory. Having too many pro-inflammatory cytokines in systemic circulation is toxic, is very lethal. Right. And my thought was IL-2 first, inducing inflammation. IL-10 is one of nature's anti-inflammatory. Let's see if we can put those together to take away the tox, but retain the potency. Again, multiple groups have proven or have published that IL-2 and IL-10 together on CD8 and NK cells is very beneficial for the cytotoxic circuit. And then lastly, at ARMO, we found that when you dose cancer patients with PEG10, you actually block Treg expansion. So the other liability of IL-2 is the induction of proliferation of Tregs. So if it's true then that CRS is the real toxicological issue with 2, and we can add 10 to take that away, and 2 plus 10 on CD8s and NKs is better than either alone, and we can block that toxic or that efficacy limiting induction of Tregs, then theoretically we should have a compound that in patients whose immune systems are kind of sufficiently capable of responding, we should see anti-tumor function. And moreover, by concentrating it in the tumor microenvironment at the tumor cell surface, we should be able to really help T cells recognize tumors. So all of that seems to be true. We've dosed 27 patients. We have stable disease thus far in a number of folks that have cold tumors. We're just now starting to dose the kind of the real patient population, which is RCC. Both molecules scored in monotherapy in RCC, and RCC is a 2 log higher expressor of the EGF receptor, which is our current target, than any other tumor. So that all seems to make sense. We see T cells proliferating. We see, good biomarker that makes sense. We see gamma and nothing else. So we definitely have protected against CRS, and none of our patients have vascular leak either. So we kind of have proof of concept, and now we're literally just looking for tumors to start shrinking in context of this molecule. Now, the interesting thing about unleashing a person like a protein biochemist like myself into the lab is we've also proven that IL-10 takes away the inflammatory toxicity of basically every other cytokine. So IL-2 plus 12, IL-10 plus 12 takes away the CRS circuit, interferon alpha, IL-7, IL-15, kind of you name it, we can take away that toxic liability. So presumptively, we may actually be able to harness the promise of all these different cytokines. And the reason we're doing it is that, going back to grad school, I needed a source of T cells. And so I'm, a walking source of T cells. So I would take them from myself. Well, as it turned out, my buddy was a walking source of T cells as well. So I would take them from him because I'm just a faster runner than him, even with my busted leg. So the interesting thing is has always been a great responder to IL-12 and really no other cytokines. I don't respond at all to IL-12. I respond to IL-2 and IL-10 and kind of not much else. So from the beginning of my interrogation of the immune system, it became clear that we don't all respond the same way to the same cytokines. And in fact, COVID taught us that we don't all respond the same way to the same immunological stimulus either, relative to inflammation, et cetera. So the other half of the equation is mapping the human immunome in terms of how we all respond differently to different pairs of cytokines, because the idea is that it may be true that part of the reason there was only 15% ORR to IL-2 and 25% ORR to PEG-10 is that, those other folks that weren't responding, it wasn't so much the tumor, it was their peripheral immune system wasn't designed or selected to respond to those cytokines. And that's actually what our data suggests, because we've looked at about 400 different healthy donors assessing the response, kind of gamma proliferation response to different pairs of cytokines. And it's abundantly clear there are folks that just don't respond to given pairs, but do respond to a different one. So the other half of the equation then is not just, proving that 10 takes away the toxic liability of a potent cytokine and retains potency. It's also then to figure out who is who will respond the best to a given pair of cytokines, and then we have the IP, et cetera, kind of built around enabling us to do that as we go forward.
Yeah, you definitely want to stay on Deka, but it's evident you've come from humble beginnings. I personally also grew up building fences with my father and family, and I feel like a lot of those reps and, kind of level of creativity where you start from kind of a blank canvas and you build something and you're proud of it and you can kind of stand back and see where you went with it. I mean, end of the day, biotech is an art, just like a painter has a paintbrush. And so, one thing you mentioned that I think is important is when you were back in California, you were doing your initial research around IL-10 and others. You mentioned that despite all the white papers, all the scientific research, all the other endeavors from Nobel laureates or whomever else, like they said, you're wrong. This is never going to work. And you just kept kind of pushing through. And I think that resonates probably with a lot of scientists.
Yeah.
So can you just speak to that part of your career and how you essentially address those naysayers and continue to push forward, and then also how you mitigated some of the risks that I would assume were more pronounced during that time?
Yeah. So I'm a contrarian by nature, and not contrarian in your face, like I'm right, you're wrong. Not at all. And in fact, this was an argument that I had with Liz Grimm in grad school, and she said, "John, you have to form a hypothesis." And I said, "Liz, I'm too stupid to form a hypothesis. I don't know enough. What I'm doing is testing a system. Here's a system that I perturbate with a single factor, and then I ask, what has it done? Right? I let the science lead me, as opposed to me leading the science." And so the way that I speak to naysayers is, first and foremost, I only speak to data. I never hypothesize. I never pontificate. I never push what I think it should be doing. It's rather, okay, it's a Boolean logic string. If then, if then. And when you build enough if then statements, you, just as you said with regard to painting a painting, I think of it more maybe in a digitalized sense of pixels. But similar to digital versus analog, analog is 1-to-1. Digital, which is how the immune system works, is 1-to-many under the right conditions. And so to me, it is literally all about data. And the more pixels that you can put into that image, the clearer it becomes. And I mean, true refinement of, I don't know that that's true yet. So build a testable experiment, right? And test it and see. Right? See where the data leads you. Because, even with, say, PEG-10, I found that inadvertently, completely by what I call stupidity, that IL-10 on T cells activated them to be more cytolytic. I thought I was wrong. So, and then it was true. And then it was true, and it was true, and it was true. Right. So that's part of it. The other part of it is, for naysayers, I don't know any other way to answer this. To be honest, I grew up super poor, and I think what you learn when you're poor is that other people's opinions don't matter.
Right.
And you learn, it's not that you ignore someone's opinion, but you take it in stride and you say, okay, that's your opinion of me at this given time, or of the data at this given time. I hear you. I also hear the implicit opportunity. If you're saying something is particularly wrong, well, that's an experiment that I can do to see if it is or isn't. Right. So I think I started, engaging in what I guess we now call active listening at a very young age to really understand, is this an issue of me or of the data, or is this that person's issue? Right. Because sometimes you have people that just sling arrows just to sling arrows, but not all the time. And to me, the reason I love science as much as I do is that it is a discourse. It is a notion of building on that which has been before. And if suddenly you get to a place where the data isn't making sense, well, it means you're missing something. Right. And that missing something enables you to go, I'm very quixotic. You go on a search, you go figure out what it is. And that was, early on when I did nerve damage to my leg, and I was paying for school. Right. And I thought, okay, I need to make this worth something. I'm here, I left home. I'm here. I could go, build houses again, but I have to make the reason for why I'm here. It has to make sense. Right. What am I doing? Well, I know myself well enough, and as it turns out, other people have kind of cottoned on to the same thing as they worked with me, that I don't sit still. I'm not one to take anyone at their word per se, especially when it comes to science. And I want to do something new. So from that perspective, when I was thinking about a career path, the reason biotechnology seemed so interesting and, not staying in my hometown was that it was this optionality of a completely blank canvas to go do something that no one else had done. When I was little, the thing that bothered me the most was when somebody said there are no new thoughts under the sun. And I was like, what? That can't be true. right?
Yeah.
But you move yourself into a place where suddenly, as Mark said, he's the first one there. He's thinking new thoughts because no one else has ever had that system to think about. And so that's, to me, it's, those are all hand in hand in the sense of the quest for new knowledge, the filling in of the data, the filling in of the image, so that when you have naysayers, hopefully you've thought about your system more than anyone else. And if somebody asks you a question, you have an answer. Right. And that's partly the value building in biotech is that is very binary in the sense that when you get to diligence discussions, you better be able to answer 99% of all the questions that they have. And if you can't answer 10%, you say, that's a great question. This is what we're doing to address it. Right.
Right.
And then if they get to a place where they ask you a question, and you're like, I absolutely have no idea, then say that. Right. That's the other thing I love about science is that it's true in the sense that these are the facts that I have. This is what I built on. And this is what we're trying now. So,
yeah. There's definitely power in saying no. And there's power in having the ability to do additional discovery. But you surrounded yourself by incredible mentors, along your trajectory and arc of your career. And I don't think we have time to, give a level of respect and sort of, overriding due diligence to what each of them have done for you as an individual and as a leader. But within that regard, as a leader and as a manager across, your track record in pharma, biodiscovery, now leading, Deka and scaling that company, what are your guiding principles then as a leader, as a manager, and how you're building the culture of your team?
So first things first, like I tell my kids, 90% of life is figuring out what not to do. And I, it sounds flippant, but I think understanding that you don't have all the answers all the time, and that no one expects you to. There's a presumption of ego, especially in you're a founding CEO of a biotech startup. You must have a big ego to take that risk. Not so much. It's not about ego. It's about curiosity, in the sense that we have an opportunity for the first time in so far as we know in human history to actually de novo create molecules, right? That don't exist in nature,
right?
Together. And again, the barriers to entry 20, 30 years ago were categorically different than today. I mean, my labs today have more toys, more instrumental toys to interrogate what we do than most academic labs, multiple labs, and we do that for a reason. And it is purely predicated initially on an insatiable curiosity to see what's around the corner, to understand through analysis what this means. So that's the first thing. The next is, you've got to be passionate. I mean, doing what we do is a labor of love. We get paid, and there's an opportunity to sell a company and all that kind of stuff. But I can tell you, money is it's not useless, but it's not interesting, to be honest, right? And so if you're coming to work for money, you need to go not work at Deka. And in fact, I have this routine where I get really, really amped and really excited because I can't help it. When I'm talking about new data, when I'm talking about how this came to be, et cetera, it's infectious to me. Like, I wind myself up. And so we had a couple new people start, and I was doing one of my team meetings, and I said, guys, like, if this doesn't turn you on, then this is not the company for you. Life is too short. Follow your passion. If your passion isn't here, go somewhere else, right? And not offensively. Like, I am giving you everything I have, and I would like you to feel so excited that you're willing to do that as well, right? That exchange. And so got done with the meeting, and then I'm sitting with Pavel, my co-founder, and Chris, my corporate counsel, and this young lady comes in, and she has her computer, and we all think computer's busted. We'll get her a new one. And she goes, I don't have enough gas in the tank for this company. And And Pavel was the first one to say, wait, huh? Hold on, what? Because I still wasn't processing, right? And she goes, yeah, I mean, I heard John. I get it. You guys are passionate. You guys are curious. You guys are fired up. I totally get it. It's not for me. And I was like, okay, thank you for letting us know. I mean, she'd been on premises for like 2 hours. So,
Yeah.
and I told everyone, so she left, and then I brought everybody back into the room. I'm like, okay, guys, so this young lady, she left. This is the first time in 30 years of me getting excited in front of people that someone actually took me up on my offer of, if you're not passionate about this, you need to leave, right? So you gotta be careful, right? 90% of life, figure out what not to do, right? So you gotta kind of temper that. But in terms of our culture, I have one-on-ones. We got 36 employees. I have just revolving one-on-ones. I'm always talking to them. We're always figuring out what the data means. I'm very transparent about what we see in our patients, as much as I can be with regard to what we saw in a dish, in a mouse, how that all goes together. I'm very transparent about our board meetings, about raising money, how much money we have, what the environment's like, et cetera. I am trying to educate this crew across the entire spectrum of what it is to be in biotech, because it's my life, and I absolutely love what I do, and I think that comes through, the Monday meetings and et cetera. I think it comes through in the sense that it's not just about the science. At the end of the day, it's patient outcome. At the end of the day, it's making people. We will never know them, but it's making their lives better. And if we can do that, once, it's a big deal. If you can do it multiple times in a sensible way, you've changed the trajectory of the species. And I tell the guys here, I'm like, guys and girls, look, I'm talking to you, but we are talking to the world. If this works, right, this is a paradigm shift. And you guys are doing it. It's not just me. Like, we're doing this together. Everyone has a part to play. Everyone is contributing to this. And that's why you all have options, because you are all owners of this company. Right. So I do everything I can to engage their curiosity, their intellect, let's call it their better nature, in the sense of enabling them to see that it's possible to really love what you do and to have a pretty awesome life doing it. And that's not what I've learned from, how to say this. So I have had folks with whom I've worked that never taught me anything, but from whom I learned a lot. And I respect those people just as much as I do the people that were actually mentors in an active way. Because what you learn in life is not necessarily handed to you; it's what you take from it. In the sense of, life happens, and it's your responsibility to be in control of your thought process, to react in a beneficial way to whatever's going on around you.
Yeah, really, really well said. And curiosity killed the cat, right? But also with that same light, curiosity cures cancer. And through your experiences at Introgen and Targenix and Armo and AZ, formerly MedImmune. I mean, a lot of that has to be kind of in your mind, reverse engineering things that you wanted to do differently and applying that to where you are now. And before we dig in a little bit more on just sort of the technology itself, really excited about the Cytokine platform and understanding that the nuances of how you're treating both inflammatory diseases, but then cancer as well, and other modalities in the future. But do you so for your employees, and do you give equity? Do you can you talk a little bit about your cap table and what your approach is to fundraising?
Oh, well, that's a lot to unpack there. So first, yes,
sure.
everyone gets equity, and it's commensurate with their kind of level in the company, their years of experience, and at every year, end of year, we evaluate folks for their contribution to the company, and then we, everybody gets a raise. The magnitude of the raise is dependent on kind of where you fall in the contribution that year toward the goals, and then as well the options that we grant. So it's the compensation committee as well as myself, is again predicated on that kind of same metric. What did you contribute to the company in terms of achieving goals? and what did you contribute to the company in terms of facilitating, developing our culture, right? So we take both kind of all of those things very seriously. So yeah, so everyone has options. Fundraising is the hardest thing I've ever done, I can tell you. I've had 3 kids. I've been married for 30 years. This is my 3rd company. Yeah, it's the hardest thing ever. And it's unfortunate and fortunate. And what I mean by unfortunate is there are ideas that are worthwhile, that have good people around them, that in this last 2 years, unfortunately, have not gotten funded. And the fortunate part is that there are ideas that maybe are a bit too far right now, that aren't getting funded. And that's probably a good thing. There's probably needs to be some maturation in the space in order to have a meaningful value proposition in a meaningful time frame. We are one of the kind of few at this point that made it through the valley of death over the last couple of years. And again, the approach to fundraising is frankly the same approach that I have to building the company. It's all about data. And it's interesting to see the maturation of VCs with regard to what that means, right? So, it used to be, even with Armo, it was more or less enough to get into patients and show that you didn't kill them, through the safety of phase 1. And we had a couple of responses in RCC in that study, and then we did an expansion cohort in RCC, and we started to see some efficacy. But the efficacy was not required then in the phase 1, and it is now. So that again is the maturation of the space. It's the focusing of the space. It's the increase in knowledge of what a real signal looks like, right? So just like I'm filling in pixels for my imagery, the VCs are doing the same thing, right? Everybody is. Our brains are pattern recognition systems. So again, good and bad. At the end of the day, it's all about generating the data to the next milestone. You have to be very careful about how you deploy capital as well, right? So we've raised about 61 million after this next close. Our nearest peer has raised about 300 million. They've dosed, call it 18 patients. We've dosed 27. It took them 2 years to dose that. It took us a year. So we're capital efficient. We are moving quickly. And the only difference is that we have a bunch of stable disease in cold tumors, and they only focused on immunogenic tumors, and they have, a couple responses. So, which strategy was better or worse? Ultimately, if I get a couple PRs in the next 6 months, then, we're good. And that's just kind of where we're at. We had to go quick. We had to deploy capital effectively, efficiently. And we also had to move quickly. And so my CMC guy always tells me, he says, you can't have speed, cheap, and quality at the same time. And I'm like, no, Dave, that's the wrong answer. I need all 3 of those all the time.
Yeah. No, it's interesting, your perspective on that. And so I want to ask you this question just within the lens of like your facilities. Deka started at the Germantown incubator, grew organically in that space, and then recently, leased a new facility in the I-270 biotech corridor. But as it pertains to the conversations you have with your venture partners, right? Facilities being such a large just line item in general, when you take into account, FF&E, the OpEx, just paying the bills to keep the lights on and all the equipment that is required. What are the conversations with VCs as it pertains to sort of the facilities piece, the hard infrastructure piece? And, a lot of that right now is companies trying to extend their cash runway. So it's, giving back space, reducing headcount, extending that cash burn, that burn rate so you can get to that next round of financing. But what were the conversations with your VC partners in particular when it came to discussions around graduating out of GIC?
So the other reason why I moved here is exactly bricks and mortar. And so the cost for bricks and mortar here is about 20% of what it is in the other big biotech areas. So 20% of what it is in Boston, Kendall Square for sure, and definitely what it is there in South City in the Bay Area. So the conversations I had were really easy, in the sense that I said, okay, we need to have a more contiguous space.
Mm-hmm.
That permits us to have everyone, kind of not wandering in and out of, these different disparate rooms, even though the GIC was amazing. But it's designed to be modular for smaller companies. And we grew to the point where we were inhabiting, I don't know, 2/3 of all the labs.
Right.
And, that's great, but it's easier for kind of the continuity of data flow if you move into a facility where the rooms are essentially purpose built for use.
Right.
And that's what we did here at Minkoff on Seneca Meadows. Is just we literally just moved across the street. And then the other thing we did is we took some space, exactly to your point, from Trinomics. They have a mouse house, and so we took over their space because they're not using it. So it's just kind of this. We were already, from a bricks and mortar perspective, far less of a burn than anyone else. Minkoff did an amazing kind of deal with us in that we amortized a bunch of the cost over a 10-year lease, which is typically what you do. But again, the price point was so much lower that hit on the bottom line of the burn rate of the company is almost a non-entity to my investors, because they're used to just these brutally high numbers for just keeping the lights on.
Yeah, I mean, we've certainly at Scheer seen a lot of creative deal structures where, working with tenants and able to amortize an additional TI for improvements. Obviously, kind of a standard build out could be anywhere between $300 and $400, or, if you look at AZ at their new space on Medical Center Drive, upwards of maybe, $1,000 or $1,300 per square foot. So, it's certainly a way that we've advised clients and tenants, specifically in this market and beyond, on just how to kind of leverage that landlord as a bank, in a sense, and being able to really manage your cash flow and operating expenses. But just diving back into your platform, right? I think that's really the meat of what we want to know about, just in terms of Deka. You kind of grew and sharpened your proverbial sword in kind of the mid to late 90s, where targeted immunotherapy was sort of this really large idea. And fast forward now in 2024, cell and gene therapy, precision medicine, and specifically precision therapeutics is really gaining a lot of momentum, just given some of the advances in the industry. And so your pipeline is really understanding just sort of the combinations of cytokines, understanding how those can treat specific patients with precision. And so, in a sense, you're radically changing what is, immunotherapy and developing personalized treatments, which I think in 10 or 20 or 30 years really could be the standard for the industry. And so can you just give us a little bit more idea about your pipeline at Deka?
Yeah. So there's we come at it from three ways. First and foremost is the blending of natural cytokine biologies that have already been proven in humans, right? So IL-12 has been in people, and interferon alpha has been in people, and 2 and 10, et cetera. So that's one component. The other component is this precision medicine aspect of not all of us responding the same way to the same stimulant. So then can you build an assay? If it's an assay that's reproducible, can you then find the genetics that might actually confer the capacity or incapacity to respond to a given cytokine? And then the third, which to me, and again, maybe this is because I am too curious and I want to see around the corner too far, is cytokines, in basic biology are called signal 3. So signal 1 is T cell receptor engagement. Signal 2 is co-stimulatory signal transduction, and signal 3 is denoted as cytokines. So that kind of means that they bridge the gap in a lot of different ways with regard to activation or even fine-tuning of what T cells and other immunological cells do. So if that's true, then just as you alluded to with regard to CARs, CAR Ts, TCR-Ts, TIL therapy, cell therapy in general, then it makes sense that our molecules may also augment other platforms particularly well. So the third arm of what we do is to evaluate the combinatorial use of our platform molecules with either cell therapy or with BiTEs. And so the reason for that then is, what are the two ways that you can modulate a therapeutic index? Well, you can take away tox, or you can improve potency, or you can do both. And so what we found our Diakines do is both. They both take away the toxicological output of a BiTE interaction, and we're seeing if that's also true with different CAR Ts. When I was at ARMO, I already knew that IL-10 would facilitate CAR Ts. And before I left, I got the team working on a combination approach of a CAR plus PEG-10, and they published on it. And in fact, it does work. So not a great leap of knowledge there. It's a cytokine that affects T cells, CAR Ts, or T cells. It ought to work, and in fact, it does. So again, take away tox, improve potency. So exactly to your point of precision medicine, ideally, it would be an off-the-shelf CAR that you could then perhaps add an off-the-shelf Diakine and give it to a person. And now you have a packaged deal where you're driving specificity predicated on the CAR, and you're driving enhanced potency and reduced toxicity predicated on the Diakine. And so if that's true, and this is new data that we just generated, the Diakine would probably have to be in the same context of how the CAR is interfacing with the tumor, right? Because most of the stuff is right around the cell-cell interaction. So we asked ourselves, we said, is the Diakine in the immune synapse? And I can tell you unequivocally that yes, it is. So similar to what Mark Davis did, he and Christoph Wolfing built the first microscope that could essentially visualize T cells functioning in real time. And they were some of the first ones to work in the immune synapse, visualizing the immune synapse. Christoph has subsequently built an entire lab around that. So, circle all the way back to my master's, I am now in the immune synapse with this molecule. And again, this is stupidity. This was not a structure function relationship that I built into the system. But as soon as I started seeing where the data was taking us, I realized that we probably had to be in the synapse. And we literally just got last Thursday, one of my awesome scientists gave us, the proof is in the pudding, as it were T cells, tumor cells, the label of the Diakine, and it's right there in the immune synapse. It's some of those beautiful data I've ever seen. But again, these pixels that we build, how do we use the knowledge that we've already generated, and then how do we jump ahead and make it even better? Because again, ultimately, it's all about patient outcomes. Patient outcomes are going to be driven by folks like me and my team and other folks understanding the combinatorial approach. Because remember, cytokines work together, cells work together. We have to become experts in that dance in order to provide true therapeutic potency for these devastating diseases. And I think that if I had $1 billion, I'd do everything. But I don't have $1 billion. I have a lot less than that. So I really need to focus our team on those critical pieces that are the most marketable with regard to Deka and the rest of the world, my audience, which is, pharma and BCs.
Yeah. No, it's really well said. And you're talking about pixels, right? And coming together for a common picture, and a little bit of community is similar, right? You're bringing all these sort of disparate pieces together. You're finding talent, you're finding ideas, you're building that ecosystem out. Certainly here in Maryland, it's evolved to be a top 5, market for it. And to your point, just from a cost perspective, substantially less than some of these core gateway markets that you think about in Boston, Cambridge, San Francisco, and others. But, Maryland scientific community is sort of unique in my mind, because you have a very robust K through 12 system, a highly educated workforce of folks pushing, their children to be scientists, similar to how your high school professors also were pushing you. And so what does the Maryland community mean to you having moved here? And, if you were to hypothesize on just sort of the evolution and trajectory of it in the future, where do we go?
I think Maryland is really extremely interesting, right? So, I grew up and then spent a number of years in California, which I always call the frothy edge of everything. And then I did my PhD down in Texas, which is the kind of non-frothy edge of everything. And right, so I had this very interesting polarized view of America. And then my wife grew up in Soviet Russia in Moscow, and so that's a whole other view of things. And we never really quite fit, and because we're kind of centrist, to be honest, because I think science teaches you to really kind of keep your mouth shut until you know what's going on.
Yeah, yeah.
Right. So moving to Maryland, it's that blend of people that are extremely educated and extremely thoughtful about what they do and how they do it. And we've really found kind of our forever home, as it were, in the community that we have here. It's a significant, a lot of expats from really all over the world. Just in our neighborhood in Bethesda, my wife has met people literally from everywhere. When she goes for a walk, invariably somebody comes out to just go for a walk with her, to walk the dogs, just to talk. And I think that's kind of the beauty of this area, is a common sense of service, right? You've got a lot of folks in the government. My daughter is at Army Research Labs. I mean, she's all about service. You have folks that are highly educated. You have folks that are wanting to push the boundary in their own thought process, and then in their children as well. And I think that's that kind of, my son went to Whitman. He's now at IU. I originally had him at Bullis because when we moved here, I wanted a smaller school. And he goes, "No, Dad, I'm a big school guy, right? I'm not a small school. I'm a big school guy." So he goes to Whitman. He goes to IU, a massive school. He's, happy as a clam at high tide. My youngest is at Bullis, and she's a super small school person. And so it's kind of that availability of really the entire spectrum of breadth of knowledge, breadth of engagement, et cetera, is kind of unique here. And to me, it is more centrist, and I really like that. I'm not saying one or Texas is better than California or California is better than here. It's just for my wife and I, we really feel at home here. And that, I think, is translated into my ability to kind of build the crew that I have, incredibly gifted young scientists. Our research core is phenomenal, and I've been able to attract folks like my CMC guy. I worked with him in Oklahoma. His wife is actually from Oklahoma City. He moved out here to work at Catalent, and now is working with me. Some of my first co-investors are folks that I knew from past lives that moved out here, and then I've met another. My corporate counsel grew up just over there in Germantown, went to GW. Just kind of this homegrown talent that has a huge breadth of experience, both from working. So my co-founder was in BD and supply chain there at MedImmune. His mom was in analytics at MedImmune. His dad was at USDA, and they're Armenian, just super educated, super engaged, just absolutely there's a genuine desire to make the world, not to sound corny, but to make the world a better place, to really be about service to the broader community, and not just the US, right? I mean, We're in the US. I love it. It's cool. It's awesome. But, we're a species. We live on a planet. And I think that the more we can understand how to engage with each other, the more we can ameliorate disease, the more time it gives us as a species to kind of sort some of the other problems of what it is to be human, frankly.
Yeah. That's something that we think about often, right? Is how do we ensure that we continue to progress in the ecosystem, be valuable partners? But at the end of the day, it's just organic collisions, collaboration, and being part of a larger community. So, really glad that Deka has sprung roots here. And, we working with BioBuzz in particular on their awards. And, Deka and yourself have been nominated as a finalist. And so what does that mean to you to be recognized in that regard?
I think it's great. I mean, from the perspective of anything that I can do to grow with this community, right? So, as you said, we're kind of top five now. To be honest, not to cast aspersions on anyone else in any other environment, but I know what the Boston scene is like. It is amazing, but it's also kind of groupthink, kind of in an echo chamber, right? And that's that. I think that we don't have that here, which I think is important. But being able to be a participant in this economy and in this ecosystem as it grows, from a small company perspective, right? Because obviously MedImmune put a lot of energy into the ecosystem as well, but that's a very different. That's a kind of a buy side. That's kind of a bigger, sibling or parent even, growing things. Being one of the little guys, and growing stuff and helping my team understand the impact of what they do, how we're engaged in our community, and then how that grows from there. It's super exciting. It's really cool. And thank you for the nomination. It's really nice.
Absolutely. And nomination is community nominated. So, it's peers voting for other peers, which is an incredible opportunity with BioBuzz. And, again, really excited to connect with you, John. Deka Biosciences, developing targeted systems for each patient with precision, leveraging 30 years of experience. And we're just really, really glad that we've had the opportunity to connect with you today on BioBlast. And, any last words for everyone?
No, thank you. Thank you very much. And, just keep following your passion, guys. It's the only way we're going to get around the corner.
Yeah, absolutely. Well, thanks, John. I really appreciate your time.
Cheers. Thanks.
Thanks.